would like to thank Kerstin and Mark and other organizers of the meeting for giving me a chance to show you an overview on SMC complexes. So I will tell you about the architecture of SMC, structural maintenance of chromosome complexes. So you already heard about cohesin and condensing, and these complexes are highly conserved. So you can find SMC-like complexes in bacteria. In fact, you can find either one of these two, or even two complexes. So usually you have SMC, SCPAB complex in bacteria, or MUC-BEF combined with this MKS-BEF complex in some of them. So in eukaryotes, most of the eukaryotes contain cohesin, condensing, and SMC56. Only some ciliates miss this SMC56 complex. So I would like to show you, and you can already see similarities between these complexes, bacterial and eukaryotic ones, and some differences, of course, as well. So, and their implications for these machines, which basically are responsible for the loop existence or loop extrusion in the cell. So they are composed of always of two SMC proteins. What is important here is that these SMC proteins have this kind of ATPase cassette, ATPase head domain, so-called head domain, which is able to bind ATP molecule and hydrolyze it. And this is actually the force which is responsible for the loop extrusion. You can also see this very long arm, which is composed of two very long helical segments, which make a coil-coil structure, antiparallel coil-coil structure. And here is the hinge domain, which is responsible for the dimerization of the two SMC proteins. They can dimerize also through alignment of the arms as well, but it's only when the ATP is not present. When the ATP binds, and actually each of the heads binds ATP and also dimerizes the heads. So upon this dimerization, upon this binding of ATP, two ATP molecules, you get the heads breached, first of all, and also you get a quite substantial change of the conformation of these arms. And actually this kind of ring was proposed to bind two DNA molecules in kind of topological manner. So the DNA is, well, it was proposed that DNA is kind of trapped inside this ring. And this, for example, in this way, for example, cohesin can hold sister chromatids together, responsible for the cohesion. Or supposedly, these two DNA segments can be the kind of the stem of the loop. But I will discuss this case later on. These two SMC proteins are crucial, but you also need this so-called Kleisin molecule here to get really a complete core of the complex. This Kleisin molecule kind of locks and finishes, let's say, the ring structure of the complex, of the core of the complex. And what we realize is that there are some similarities and differences between different SMC complexes. So, for example, here you can see that prokaryotic Kleisin molecules and Kleisin molecule of the eukaryotic SMC56 complex, they are short, while these cohesin and condensing Kleisin molecules are pretty long. So just to make it clear, these Kleisin molecules close the ring in the way that the N-terminal part this helix-turn-helix domain binds the neck of one SMC molecule, while the C-terminal wing helix domain, here, this part, this domain, binds from the bottom of the head domain. So here are the arms, here is the head arm. So this is the neck, this is the bottom. But this is common for all the Kleisins, but these Kleisins differ in the length of the linker region which connects these two regions, these two domains. And you will see it will become important on the next slides. This linker part, middle part, is always associated with some regulatory subunits. So in bacterial proteins complexes, you can find this kind of subunits, which we realized are very similar to these subunits NSE1 and NSE3 in SMC56 complex. They are not similar by sequence, but they are very similar by their domain composition, by their structure. So they contain so-called wing helix domains in tandem. And we call this new family of subunits of proteins Kite as a Kleisin interacting tandem wing helix elements. So, and we realized that they are similar. So it might be that these bacterial SMC complexes are similar to eukaryotic SMC56 complexes. And later on, our colleagues from Oxford realized that it pays also for this regulatory subunits of cohesin and condensing. They found that they contain so-called heat repeat motifs, all these subunits, and established new family of Hawk proteins for heat associated with Kleisin. And so, based on this, we propose that during the evolution, these cohesin condensing complexes evolved in some way through the SMC56-like complex. But there is one paradox in this evolution. When you look at the SMC protein sequences of the cohesin and condensing, SMC1, SMC3, SMC24, these are the cohesin condensing SMCs. They are much closer to bacterial sequences than the SMC56, so which should be somewhere between them, of course, if the evolution went through SMC56. So it was kind of conundrum, kind of puzzle, but very recently analysis of archaeal SMC proteins bring this kind of new piece to the puzzle, where most of the archaea contain SMC proteins, which are again similar to these cohesin condensing bacteria. But some of them also contain second SMC complex, which is composed of SMC proteins which are much similar to SMC56 than to cohesin condensing. So in other words, during the evolution, there was a duplication, and simply two SMC complexes coexisted in this ancestral prokaryotic organisms, with the canonical SMC and SMC56 related complex. And from this canonical one, cohesin and condensing complexes evolved with their Hawk proteins, while SMC56 complexes evolved from this SMC56 related one. So in latest eukaryotic common ancestor, there were already all these three SMC56 complexes, probably. So very recently there were developments in cryo-EM, and now we have structures of several of these SMC complexes. Here you can see one of the bacterial MAC-BEF complex, or its part, its head part. And I wanted to demonstrate the similarity between this bacterial SMC56 again on these structures. So as I told you, the Kleisin molecule binds, its N-terminal part binds the neck here of the SMC protein, and the C-terminal part binds from the bottom. But surprisingly, it doesn't only connect the simplest way here, the head domains. But you can see here that the linker really goes upwards, and with this DNA molecule in, you can realize that this Kleisin molecule here simply really locks the DNA here in this compartment. So the part of the compartment is also these are the Kite proteins which associate with the linker, and together they form a top part of this compartment. And as with other SMC and RAD50 actually as well, proteins, the bottom of this compartment is formed by the heads. So hopefully you can appreciate that this SMC56 structure is very, very similar. So again, you can see this SMC proteins, DNA at the top of these SMC heads, and again, Kleisin molecule bound to the neck, to the bottom, and here again the linker going again up and locking in the very similar way, locking the DNA in this compartment. So this nicely corresponds with what we proposed, that the bacterial and eukaryotic SMC56 are very similar from this point of view, at least here. What about the cohesin and condensing? So because they have different Hawk subunits, they have much longer Kleisin linkers, but when you look at this cohesin structure, you can again see something very similar. So SMC proteins with their heads, DNA on the top of these heads, again Kleisin, N-terminal part here, somewhere here, the linker threading through, and here is the C-terminal part bound to the bottom. And so in a very similar way, the Kleisin linker is bound here by the Hawk hit repeat protein here, which again locks the DNA here in this compartment. So surprisingly, in cohesin and condensing, you have this kind of compartment instead of just simple ring which would embrace the DNA segments. So there is a common mechanism probably, or some common structure at least, which looks like this, which simply locks one of the strands of the loop during the loop extrusion. So, and more surprisingly, from this paper, very recent paper in Science from Christian Herring lab, they also showed that not only this first segment is locked in this way here, but that the other DNA segment is probably not within the ring, but somewhere with the other Hawk subunit bound somewhere along this long Kleisin linker. So this really questions the original proposed model for this kind of topological binding, this kind of mechanism for the loop extrusion. This still pays, is still valid for the cohesion for the holding of two sister chromatids, but the mechanism of the loop extrusion might be simply different. So this is something to take into account when you, for example, do your TEF cleavage experiments, because they were originally designed for this kind of topologically embraced DNA, not for the loop extrusion mechanisms. So this is something we can discuss later. So in the summary, I would like just to conclude simple things for you. So SMC proteins evolved from some ancestral canonical and SMC56 related complexes. These complexes, these ancestral complexes, contained short Kleisin linkers associated with Kite proteins originally. And then cohesin and condensing complexes evolved from this kind-containing complexes by replacing these Kite proteins with Hawk proteins. But at the end, they all lock DNA in a very similar way here in this kind of compartment composed of head domains, Kleisin, and Kite or Hawk subunits, which do really bind physically to the DNA and keep it, hold it there in this compartment. So at the end, I would like to thank my colleagues from my lab and, of course, collaborators from Sussex University, Lausanne, Heidelberg, and, of course, other colleagues as well. And I thank you for your attention.
Okay, thank you so much for the amazing talk. I wanted to ask you about the head Glycine kit compartment. It was encircling the DNA. Do you think it's a flexible structure with the Glycine, so also nucleosome can go through during the loop extrusion process?
Sorry, what was the question?
The head Glycine, it was encircling DNA of only 2 nanometer diameter. So how nucleosome can go through during the loop extrusion process?
So clearly you need Nike DNA for this binding, for this interaction. Actually, our data and other data as well suggest that the SMC complexes prefer sites without nucleosomes for the loading, for example, at least. Yeah. And so it's not clear what this compartment does, but I can imagine when you extrude the loop, you need an anchor which is simply fixed, and then you extrude. The other part is flexible. So this compartment is probably the anchor, and the other part, if I go back, so you have space enough here to really embrace whatever.
Thank you so much.
Very likely.
So, thank you very much for this nice talk. No need to apologize that you didn't show tats or stuff. That was really, really nice. I have a question regarding the structural consequences, though. I guess someone has tried to knock out those Kleisins or Hawk and Kite proteins. What's the functional consequences? Are those essential genes or essential proteins? What happens to the loop extrusion?
Yeah, sure. As far as I know, deletion of any of these subunits, doing myself or in SMC56 is lethal. Causing condensing as well. Mm-hmm. I can recall the yeast phenotypes, for example, when you delete cohesin or condensing and you do tetrate analysis, you can see that one cell, the spore, dies immediately. While in SMC56 deletion mutants, you can see microcolonies, but still they are dead early after the dissection. So Yeah, and definitely there are data on mice and so on, clearly showing that they are essential for life.
Thank you for your beautiful talk. So I was wondering, given these beautiful structures that you show us, so do you think supercoiling might be able to go through those interactions? So yeah, so if you generate positive supercoiling, well, depending on the condensing, as shown by Tatsuya, for example, would you guess that supercoiling can go through, I mean, the DNA?
As I told you, these arms are 50 nanometers long. So this ring is big enough to embrace anything starting from supercoiled DNA to nucleosome. Yeah. Not this compartment. As I can see from the structure, it's really just fitting the DNA molecule. Yeah. So it depends what you mean by binding to supercoiled DNA. Not just one compartment, but generally SMC can bind. Actually, SMC56 has a higher affinity to supercoiled DNA.
Just a brief question. There are observations suggesting that the binding of cohesins or condensins, I'm not sure which one, resists about 2 molar salt. Do you resist 2 molar salt? Would you attribute any significance to this? Does it surprise you in any way?
So it's, I think, 1 molar, but doesn't matter. It's still very high. So this kind of experiment was used for cohesion, I'm sure. So this kind of explained why, or was used to show that it's really not physical interaction, because physical interaction of protein DNA would probably be disturbed by this high salt. But they wanted to show that it's really kind of topological interaction, not really physical interaction. For example, in this last science paper, yeah, if you look at here, you can find that the cohesin, probably entrapping the DNA inside the ring, cohesin can really keep the DNA under these conditions, while for the loop extrusion, these conditions are too harsh. They're able to get this DNA bound probably in this way, not topologically, this way, only up to half molar salt. So there are different modes, binding modes, which survive different concentrations, salt concentrations.
Hi, thank you for the talk. As you've shown us, all of these structures look extremely similar. They all have a ring-like structures, and I think even for Archaea it's the same. And it's fascinating to me that they can do cohesion and loop extrusion, at least for cohesin. But also condensing can apparently do something like cohesion, some salt-resistant DNA entrapment. And yeah, I wonder, this might be speculative, but I wonder about your opinion. Do bacterial ones also do it? Do they have to? I don't actually see why. And what do you think, if they came from Archaea, what was the original role? Was it cohesion or loop extrusion, both together immediately?
So, was definitely not cohesion, because I think this is very specialized, during the evolution, very specialized role for the cohesin only. The common thing for the other complexes, cohesion is really just a cohesin. The other complexes can, let's start with, translocate along the DNA. That's one activity which is common for probably all of them, and can extrude loops. So this is same for all of them. It was already shown that SMC56, MAC-BEF, these other guys are able to extrude loops. Of course, they might be doing it in a bit different ways. Yeah, depending, for example, on this and other things as well. The loop length might depend, or speed might depend on these features. Yeah, hard to say. So they somehow differ, but they have also few things common. So loop extrusion like.
So you think the fact that just DNA can be entrapped, like topologically, the ring opens, DNA comes in, ring closes, is kind of an incidental thing for condensing and bacterial ones. For cohesion, it has a role, yeah, but for the rest?
It has some role, and to me at least. I don't think it's clear to the others as well either. It's not really clear how the loop is extruded, what takes part. If you look at here, yeah, you have here this compartment, but you don't have this long protrusion, long linker region, other Hawk protein which would bind the DNA. So the second segment of DNA must bind somewhere, probably inside, but it's still not clear how it protrudes, and so on. For loop extrusion, actually, for SMC56, you need 2 monomers. So it might be some kind of combination of translocation and loop extrusion together. Not fully clear.
Yeah, I agree. Thanks.